HubLE viewers, we thank you so much for joining us today. My name is Thomas Ogola. I am a PhD student at the University of Sheffield, studying in the Department of Oncology and Metabolism. Today I have with me Dr. Christina Vrahnas from the university, currently in the University of Dundee in the Dr. Ian Ganley's lab, the MRC protein phosphorylation and ubiquitination unit. And she'll be talking to us about her paper, which is here. I've got the paper here. It's increased autophagy in FNB2 deficient osteocytes is associated with elevated secondary mineralization and brittle bone. So although she's at the University of Dundee, she actually completed this work in Natalie Sims' lab in the St. Vincent Institute in Melbourne. And so over to you, Christina. Could you please tell us a bit about this paper, the background, hypothesis, your findings?
Yeah. So as I mentioned to you earlier, I was in the lab 3 years ago now. And so when I started my PhD in the lab, they were focused on understanding the mechanisms involved in bone formation to identify new pathways for the treatment of bone disease. So FNB2 was one of the proteins they were interested in, and it's a contact-dependent signaling protein that's expressed in osteoblasts and osteocytes. And so previous data in the lab showed that when they treated osteoblasts with parathyroid hormone, PTH, and PTH-related protein, they increased this FNB2 protein expression in osteoblasts and osteocytes. And these PTH and PTHRP are pharmacological agents used in a clinical setting to increase bone mass in patients with fragility fractures. So when I joined the lab, they had data showing that FNB2 was required with its interaction with its receptor FB4 for continual osteoblast differentiation. And they also showed that when FNB2 was deleted in the osteoblast lineage in mice, it compromised bone strength by impairing the initiation of mineralization. And so this led to bones that were more bendy or more flexible, which was sort of like a mild osteomalacia-like phenotype. And so in this paper, we wanted to know what the role of FNB2 was past this checkpoint that is required for osteoblast differentiation. So its interaction with its receptor FB4. We called this the FNB2 FB4 checkpoint. And so the hypothesis or the aim of the paper was to investigate what deleting FNB2 later on in the osteoblast differentiation pathway would be. And what we found was, it was quite interesting in that when we deleted FNB2 in osteocytes, they showed a completely different bone phenotype. So instead of the bones being more flexible, as in the earlier mouse model, we found that the bones were more brittle. And this was quite surprising because we didn't really expect that. And there was nothing in the bone phenotype by histology or by micro-CT that we could detect was the cause of this brittle bone phenotype. So bone mass was unchanged, the tissue mineral density was unchanged, the initiation of mineralization detected by histology was progressing at a normal rate. And so we knew that another contributing factor for bone strength was bone quality. And these mice, we asked the question, do they have a defective bone material? So we developed a novel method at the synchrotron in Australia, in Melbourne, using Fourier transform infrared microscopy. And it's a nice method to analyze really small regions of bone. And we could age match the bone regions based on fluorochrome labeling. So we knew how old the bone was that we were analyzing. And so we found that in FNB2 deficient mice, so mice lacking FNB2 in their osteocytes, had mineral accrual that was accelerated. So the secondary mineralization process had a mineral that was essentially being dumped into the matrix at higher levels than control mice. And so this is really interesting because we were learning a new method that we had no idea how to do from the beginning, but we learnt it very fast. And then we wanted to understand the mechanism. So we knew that the bones were more brittle because they had more mineral, but we didn't know what was causing this increase in mineral accrual. So we did some RNA sequencing to find any differentially regulated genes that could be causing this phenotype. And we found that a lot of autophagy associated genes were dysregulated. And so autophagy is a process, is a lysosomal degrading process, and it contributes to a lot of different cellular functions. So we didn't know much about autophagy at all. We're not an autophagy lab. And so this was a new field for us to explore. But we were able to show that osteocytes lacking FMB2 had increased autophagosomes, both in vitro and in vivo. And this was with a lot of effort from collaborators around the world, which was great. And so Martha Blank, who's the second author on the paper, she joined the lab after I finished and carried on with the paper after I left, and found that when you stimulate autophagy in osteocyte cell lines, you increase mineralization. And then she eventually found that FMB2 suppresses autophagy in a ROA-ROCK dependent manner. So this proved that autophagy processes in osteocytes may directly control mineralization. So this was quite a novel finding for us. And it also revealed like a novel mechanism for the process of mineralization of bone that is pretty poorly understood, which was exciting for us. Yeah. So also the process of autophagy is not really well known in osteocytes. So this was a new exciting finding for the field for bone biology, I think.
Okay. Yeah, so it's very interesting to hear you speak about it, because it's quite a comprehensive paper. But a lot of what I'm getting is that some of the things were actually quite new to you. You didn't know about that. You kind of went to it, or it's a new technique which you hadn't really done before. So there's a lot of novelty even for you and for your research group. So what did you find most challenging in the whole process?
I think the most challenging thing was sort of getting into there were two things. Firstly, it was developing a method to look at bone material quality, bone material, because we didn't have experience in that area, and we sort of got advice from different people. And then when we went to the synchrotron, we were working with physicists who didn't have a background in bone biology. So we collaborated a lot. And it was challenging to get results that you didn't quite understand, but you had to really make something, you had to make sense of it. And so it took years for us to really understand what all of it meant. And I was also able to have the opportunity to travel overseas and visit some labs who also use this method in different tissues. So I learned a lot about it during my PhD. So yeah, the FTIRM method is a very nice method, and that was quite a challenge to understand completely. And the second thing was, the challenge was always understanding why we had such a different bone phenotype. And it was, of course, using this method that we were able to distinguish between why FMB2 deletion in osteoblasts compared to osteocytes resulted in a different strength phenotype. And also the challenge of publishing is always a challenge for everyone, I think. So that was also hard to get the paper.
Yeah, and it's in Nature Communications, this is very good paper, congratulations. And so what was the process of publishing to Nature like? When you have your paper, you've written up your manuscript and you're thinking about where should we submit it? How was that process, and is there anything you'd like to share about so where you decided to submit in the end? And do you have any advice for people who might think, okay, I'm not sure this might quite get there?
Yeah, it was a long, challenging process to submit to Nature Communications. And I think with high impact journals, you can expect that the review process is a long one. And people should expect that when they're submitting their work to a journal like that. ultimately, the review process was beneficial for the paper in that we were able to get a mechanism out of it. So we were able to understand why FMB2 was causing, or why deletion of FMB2 was causing an increase in autophagy, and so it was a good thing in the end, and it was not easy. And I think the help from Martha, who joined the lab, was I couldn't thank her enough for finishing off the work, and also Natalie's persistence in the review process and continuously sending me updates while I started in the new lab was super helpful. And I think, the reason why we chose to submit in a journal like Nature Communications was because we knew that the work was quite novel and we thought it was really interesting. And we had a lot of new and novel methods involved in this paper, which involved a lot of collaborative work. And so we had, histologists, biochemists, physicists involved. And so we thought it would probably be a good journal to submit to, because it wouldn't just be of interest to bone biologists, it would be of interest to a broader scientific community. So we thought that would be a good idea. And I'm
glad the people at Nature Communications thought so too.
Yeah.
So, so and it's quite multidisciplinary. There's quite a few things in there, and as you said, it could benefit a lot of a wider range of people. There's imaging and things like that in there, which you developed for this course. So I know it's not all doom and gloom, making your way to Nature Communications. So what did you enjoy the most about it? Either the process of publishing or even the experiments, and I'm sure there's a lot of green spots in there.
Yeah, I think I never sort of, during the process of writing the paper and doing the experiments, I never thought we would submit it to Nature Communications. It wasn't like my ultimate goal. The positive is that it was accepted. And because it was such a long process, I think one of the highlights for me was leaving the lab and having other people do work that sort of confirmed my hypotheses was nice. It wasn't that I was the only one that saw things. Other people could replicate and show what we were thinking, which was nice in the lab. And the positive was that, obviously in the end it got accepted. But now looking back on my work, because as I said to you, I haven't been in the lab now for 3 years with Natalie, looking back and seeing what I had achieved during that time in the paper is probably the best feeling, is knowing that you did all this work and it's now out there for people to see, and hopefully to help people with their research as well. So I think the publishing aspect of science is very difficult and not enjoyable at all for anyone. I think it's the acceptance at the end that's the most rewarding feeling.
Okay, well, so at least we have a motivation that even though the process is a bit difficult, at the end, when you look back, it's really worth it, all the work and all the effort into publishing. So just in terms of the research here, what do you think is future work to be done in, now that you found the findings of the paper? What would be the next steps in the research?
I think sort of the unanswered questions that we had left in the paper was really trying to focus and really pick out how autophagy is regulating mineralization in osteocytes, or how autophagy in osteocytes is regulating mineralization. It's quite hard to answer those questions in a bone lab because we're not experts in autophagy. And a lot of papers published out at the moment have not focused on autophagy in osteocytes. And so I think some cool things would be to know how autophagy in osteocytes, and if they are releasing mineral from the osteocytes directly through these autophagosomes or these matrix vesicles that some people have been reporting in osteoblasts. So is this happening in osteocytes? And yeah, figuring out if autophagy is really a key process for osteocytes in the mineralization process. I think that's probably a future question that should be answered.
So yeah, thank you very much for listening to this interview, HubLE viewers. Again, my name is Thomas Ogola on behalf of the Meet the First Author section of HubLE, and this is Christina Vrahnas. if you'd like to get involved in HubLE, please get in touch through our various avenues and social media. And yeah, I wish you all the best, and I hope you've gleaned a lot of insight from this interview. Thank you very much.